in this third short video, what I'm going to do is I'm going to talk through question 3 of the Combined Science Paper 1 Foundation Tier from May 2018. So, question 3. It talks about a student. So it says a student investigated the effect of enzyme concentration on the mass of the product formed in 1 hour. So obviously in this experiment, what's happening is there's an enzyme, they're changing the enzyme, so this is the independent variable, this is what they're changing, and they're finding, or they're measuring the mass of the product formed. So that's the dependent variable, so this is what they're changing, and this is what they are measuring for their results. So it says figure 3 shows the results of this investigation, and this whole thing here is figure 3. Now the question, or the instruction first: complete the graph by plotting the points and drawing a line to show the trend in the data. Now that question is actually, or that instruction is actually made up of 2 parts, okay, and that's why it's worth 2 marks. So the first part of the question is complete the graph by plotting the points. The second part of the question is draw a line to show the trend in the data. And very helpfully, what they've managed to do is they've managed to draw the first 3 points that we plotted for us. So this is 1, 2, 3. Okay, so all we have to do is plot the other points. So to plot them at 20, it's 32, so there's 20, that's the concentration in arbitrary units. You go up 31, 32, and I'm going to draw a small little cross. Tick that one off as I've done it. 25 is 38, so 25, 35, 36, 37, 38. So we've got our next plot. Then at 30, put my ruler on the line, go all the way up, and it's 40. Tick that one. At 35, it's 40, and at 40, it is 40 as well. So that's the first part. I've plotted the graph to show the points. Now the next thing, I've got to draw a line to show the trend in the data. So my line, I'm going to start from the first bit of data that I've got. Let's get rid of that, and I'm going to draw a line. I'm kind of hitting all the points, and then the key thing is at the top, this line flattens out. Now I've done that really quite badly at the top, okay, and it's because I'm not very good at using this pen at the moment. So let's just try and draw that again, and that's why I need to, in your exam you need to do it in pencil. So draw a line, that's much better, and then at the top, as you can see, it completely levels out. Now what that shows me is that at this point, after 30 arbitrary units, it doesn't matter about the concentration of enzymes, okay, because after that point there's no more produced, okay, it levels out, there's no more produced, so the concentration of the enzymes doesn't affect the result. Now we're going to move down to the next question. The question states, describe, let's find the question. The question states, describe the effect that enzyme concentration has on the mass of the product formed. So what does the enzyme concentration have on the mass of the product formed? So I'm going to quickly look back at my graph, and I can see that as the concentration of the enzyme gets bigger, the mass of the product formed gets bigger, but after 30 arbitrary units, the mass of the product stays the same. Okay, so you're just telling the story of the graph. So it's going to be my sentence. So I'm going to talk about, first of all, as the concentration of the enzymes gets higher, the mass of the product formed gets higher also. Okay, so as the enzyme concentration gets higher, okay, and then I'm on to the green part of my sentence. the mass of the product formed gets, and I'm just going to double check by looking at my graph, yeah, it gets higher, it gets higher. Okay. So that's the first part. I've told the story of this part of the graph. Okay, as it gets higher, the mass of the product formed gets higher. Now for the second mark, I need to tell the story about this part of the graph. So for the second mark, but at 30, I'm going to write AU for arbitrary units because that is what they've used here. At 30 AU, the mass of the product stays the same. So that's worth 2 marks, and I've talked about both phases of the line. I've talked about this phase where as the concentration gets higher, the mass of the product gets higher, and I've talked about this phase where after 30 arbitrary units, the mass of the product stays the same. Now the next question, it looks kind of complicated, the next question. Well, let's have a look at it. So it's talking about the ratio, so it's a math skill we're looking at. The ratio of the enzyme concentration to the mass formed using an enzyme concentration of 40 arbitrary units is 1:1. Now with the question, it says calculate the ratio of enzyme concentration to product formed when the enzyme concentration is 5 arbitrary units. Now let's just first of all, let's understand this. The ratio of enzyme concentration to mass of the product formed using an enzyme concentration of 40 arbitrary units is 1:1. So let's look at the graph. Okay, so at 40 arbitrary units, I have 40 mass of the product, 40 grams of the product formed. So yeah, I understand that that ratio is 1:1. Now let's look back at the question. So the question says calculate the ratio of enzyme concentration to product formed when the enzyme concentration is 5 arbitrary units. So let's have a look at this. Enzyme concentration 5 arbitrary units. Go up on my graph, go across, and it's 15. So that ratio is 5 to 15. Okay, so I'm going to go back to my question. Calculate the ratio when it's at 5 arbitrary units. So it's 5 to 15. So it's 5, not to 10, to 15. Now, it says calculate. I've just read that off the graph. So actually what I need to do is I need to simplify this. So 5 to 15, they both divide by 5. So we're going to talk 1 to 3 as our simple ratio. So the next part of the question, question D, again there's a lot of information that's given here. So the investigation used the enzyme pepsin from the stomach at a temperature of 37 degrees and a pH of 7. Which statement gives one way to increase the mass of the product formed in its investigation? Now the key thing from this is it says pepsin from the stomach. Okay. Now 37 degrees Celsius is body temperature, so that seems alright. But I know in the stomach I have stomach acid. So what that means is maybe I need to do something with the pH. Okay, and then maybe I need to decrease the pH. So let's have a look at the options. Which statement gives one way to increase the mass of the product formed in this reaction? So we've got increase pH. Well actually no, I don't want the pH to go over 7 because it'll make it more alkaline. What I want is to decrease the pH. So that's not one of my answers. Okay, decrease the temperature. So if I decrease the temperature, I'll reduce the rate of reaction. I'm trying to increase the mass of the product formed, so it's not decrease the temperature. Decrease the enzyme concentration. So let me just look at my graph. At a lower enzyme concentration, I have a lower amount of product formed. So it's definitely not that one. And the last thing says increase the substrate concentration. Okay, so the substrate is the thing that the enzyme is breaking down. Okay, so if there's more of the materials for the enzyme to break down, then it's going to be able to react faster. So the answer is D, increasing the enzyme concentration. Okay, now the final question again, the final question in this is going to be the most difficult question from the series of questions we've seen so far. Explain why a temperature of 80 degrees Celsius was not used in this investigation. So in this investigation, so this refers to the investigation here. Okay, so it's the investigation with pepsin from the stomach at a temperature of 37. So why do we not use 80? Well, simply 37 degrees Celsius is the optimum temperature. Okay, that's why it's in your stomach at 37 degrees. At 80 degrees Celsius, what do I know about enzymes? So I know that enzymes denature if they get hot. So at 80 degrees Celsius, the enzyme will be denatured. So if it's denatured, that means it doesn't work properly. And the reason why it doesn't work properly is because the active site will have changed. Okay, so let me just check it. Explain why a temperature of 80 degrees Celsius is not used in this investigation. So I know that 37 degrees is the optimum temperature. I'm happy with that. At 80 degrees Celsius, the enzyme will become denatured. So why is that important? That's important because the active site, I appreciate my writing, it's not beautiful and neat, because the active site will have changed shape. Okay, let's put change shape at the end of that. And if it's changed shape, that means that the substrate won't be able to fit into it, okay, which could be potentially a fourth mark. Okay, so if you wrote something like that, that is absolutely fine as well.